everyone. It's my special pleasure today to introduce Dr. Kate Damme, who is an assistant professor right here at UT Dallas in the Department of Psychology at BBS. She joined UTD this year from Northwestern University, where she received her PhD, then continued as a postdoctoral fellow in the psychology department and on a P32 award at the Institute of Innovation in Developmental Science. It is rapidly establishing herself as a leader in the study of neurodevelopment, adolescent development, risk of psychopathology, and interventions to promote healthy neurodevelopment within an incredibly productive court of publications and also editorial work. In the current research funded by the NIH, she tackles significant questions leveraging large data sets to shed novel insights on the role of estrogen plays in psychotic life experiences in the transition to adolescence. And maybe we'll be fortunate to hear some of those new insights that she got today in the talk. Without further ado, help me welcome Kate.
I'm very excited to be here today. I'm going to use this as an opportunity to talk a little bit about my work, but I'm going to be talking more about how I'm conceptualizing the sort of area of programmatic research and seeing using the CDL sort of as a testing ground. So I would love to hear your thoughts on how this is being conceptualized, and really, I'm saving a lot of time at the end to engage in discussion. So I look forward to using this brain trust and talking to you today. So I'm going to start out by talking a little bit about psychosis. Psychosis is fairly prevalent. 1.5% to 2% of people across the globe have this disorder. For those who have it, it can be quite devastating, ending lives 10 to 14 years earlier than a normal lifespan. It's also associated with a high economic burden. And to put that into proportion, it's about 1.65 of the GDP dedicated to loss of productivity and care for those with schizophrenia. So it is a large burden, even with this low consistent prevalence rate. So this all has culminated into people wanting to act earlier, detect this earlier, to see if we can get in and intervene earlier before we have the substantial loss of function and loss of life. And this has been modeled in a sort of dimensional way across early development, with symptoms emerging early and increasing in intensity and specificity, with onset largely occurring in adolescence. And so these early staging and clinical risk models focus on a lot of times just near the first time of onset in this clinical high risk or prodromal period, where people have symptoms that are schizophrenic-like, but they don't meet full criteria for intensity or conviction. So you may be experiencing some odd thoughts. An example I like to use is the patient we have that said, I feel like the postman is trying to poison me, but whenever I think that, I also think, that's odd. Why would I think that? You know, never had access to my food, never seen this person except for, you know, buzzing by in his little car. And as you see that symptom progress over time, it starts to become intense, more conviction, not easily dismissed. They suddenly have this knowledge that this person is substantially trying to harm them, that's starting to disrupt their life. So it can transition from this promorbid stage pretty quickly. It's also associated with functional and cognitive decline as these sorts of odd thoughts and symptoms start to emerge. You see individuals who are starting to have these experiences, socially isolate, and have sleep and other health dysfunction. And this is often preceded by a clinical period where we see mild symptoms that would not probably register as clinically significant. So these are the types of things where, I often think that I hear someone saying my name, but then I'll check the house and nobody's there. So I move on from that experience. Or I often think that I see something out of the corner of my eye. When I go to check, there's nothing there. These individuals also often have cognitive impairment and may experience perceptual abnormalities like I just. Even earlier than that, when we look retrospectively, we see that these individuals have report having early social impairment, early learning impairment, and often motor delays or motor challenges in early childhood. And so there seems to be this early attenuated model of risk emerging and then becoming. So this is the clinical model of risk. This is how we've been conceptualizing it. This is how we've been working with it. We've been thinking about infancy, childhood, and adolescence as these relevant stages. And while this is true, this is actually mostly true for men with psychosis. This was built on a model of men. This works best for men. And women are a little bit different. It's nearly equal prevalence among women and men for having psychosis, depending on what amount of the lifespan you look at. And though onset and though it's nearly equal, onset and course differ in interesting ways. So initial onset follows, like I just mentioned, this very prototypical pathway in men. We see that we start to have emerging symptoms and start to have emerging onsets in this sort of adolescent period, with the highest risk for first onset occurring in this transition from adolescence to adulthood. But in women, we see something a little bit different, where they retain about 5 to 10 years of functional life before their first onset, and they have a delayed peak of onset. And this has been replicated several times over. We also see that they have this late life spike that we don't see in males. This is happening most often in this perimenopausal period. For those who do have psychosis, we also see reliable sex differences. This is taken from a large recent meta-analysis. And while men and women don't significantly differ in their positive symptoms, and these are sort of like the defining features of psychosis, having delusions, having hallucinations, these perceptual experiences that are added to a normative experience. We do see differences in the other areas that really define this disorder. But the most interesting one is that they have increased and better maintained functioning throughout the course of the illness. So they're a bit more able to maintain social relationships. They're more able to maintain employment and care for themselves than male. So one thing that I find very interesting about this regularity is that psychosis clinically is very heterogeneous. The risk factors that bind to elicit risk for psychosis is heterogeneous. And for each individual, they have unique risk factors that precede onset. And despite having these unique risk factors, this unique burden and symptom display, we see this really common grouping between men and women. And so it's been hypothesized that this is related to rising ovarian hormones. And we do see evidence for that across the reproductive lifespan. Many of you might know that there's heightened risk in postpartum for postpartum psychosis, and that's when related to a sudden drop in ovarian hormones post-pregnancy. You see a similar parallel during the perimenopausal, postmenopausal period, where a sudden drop of ovarian hormones is related to heightened risk of first onset. And then for individuals across the lifespan who have their first onset at other times, they're much more likely to have that first onset during the luteal phase of the menstrual cycle when estrogen and progesterone are lower. And for individuals who are after their first onset, they tend to have more new episodes and increase in symptom severity during that luteal phase as well. But I'm more interested in what is happening in this time period of sudden rise in these ovarian hormones. What is causing this delay in first onset? And how can we look at these sorts of resiliency factors that we're seeing relate to increased risk when estrogen's removed? This is kind of difficult. Puberty is something that's a little bit hard to define, and that's in part because individuals vary a lot in their sensitivity to these steroids. And so I'm going to be talking about it in terms of menarche, so when an individual first starts menses, and that's because that becomes a sign that estrogen within that person has reached sufficient levels to start other downstream biological effects. So we know that it's achieved some saturation point for their individual sensitivity. So to put this all together, we don't think that there are separate versions of psychosis. There's not a male version, there's not a female version. But instead, I think that estrogen may be acting on mechanisms of psychosis onset and symptom severity to reduce as a resiliency. And that removal of this resiliency factor then increases risk and severity for psychosis. So I think that we might be able to leverage what we know about sexually dimorphic neurodevelopment during puberty to understand and identify potential treatment targets for psychosis. Easy, right? Of course not. There's a problem with that, which is that we actually know very little about women's brains in neuroendocrinology. So this is a meta-analysis taken from Emily Jacobs' group. And this is major neuroimaging papers from 1995 to 2018. And while only 2% even report, what stage of menstrual cycle, are they pre-post menopause, these types of endocrine data, only 5 of them even look into it as a variable of interest or a potential variable of interest. The rest are correcting or excluding for that data. So we have a very large knowledge gap in what exactly is happening in these time periods like puberty, pregnancy, and menopause. This is made even worse in psychosis. So just to orient you, I'm going to take a second to explain these graphs. This graph on the left is the percentage of papers in the last 20 years in psychosis where they did not include women at all. Remember that women make up 45 to 50% of patients. These are the percentage of publications that did not include women at all. And on the right side here, we have, for those who did include women, what was the proportionality of women in those samples? And you'll see that we're not, here in high-income Western countries, we're still not even close to achieving this low-end estimate of 40–45% of women in our samples, let alone studying women-specific factors. So there's a lot of potential here for us to learn about these interesting issues and examine these potentialities. So this also, returning to this overall project, This is also complicated, and I'll admit that. It's a complicated thing to look at, resiliency. Risk factors come from all over the place. Everyone's different. So I'm going to propose a specific sort of programmatic approach. And that is, we do have known neural risk models of psychosis. So we can look at these models and see if they are being influenced in a sort of resiliency way. And a number of models seem to be good candidates for this kind of work. For example, the cognitive dysmetria theory of psychosis states that psychosis vulnerability and cognition and symptoms could be due to disconnectivity from the cerebellum through the thalamus to the prefrontal cortex. This overlaps pretty well with major signs of estrogen. And on top of that, estrogen plays critical biological roles in maturing Purkinje cells, regulating cerebellar plasticity, and impacting network coherence. Another potential theory is thalamic disconnectivity, which looks at just the thalamus to frontal areas as a potential risk factor. In estrogen, treatments have shown that thalamic connectivity is the pathway that might help preserve cognitive abilities, that estrogens impact functional networks through this action on the thalamus, and that estrogen treatments may selectively particularly support this frontal connectivity. So it may be the specific connectivity that we're interested in. There's the dopamine hypothesis, which is a fairly famous hypothesis of psychosis risk. This again is has a lot of potential for estrogen to be playing a role, as it modulates D2 receptor availability in a selective pattern. This has just been studied in primates. But we also see that estrogen levels across the cycles in humans modifies dopamine network coherence, in that estrogen shapes dopamine-dependent cognitive processes. So what can we do with this? Well, we can take a normative neurodevelopmental approach. We often do this in other areas of science where we create this sort of normative pathway or normative developmental course, and we look at alterations. And we've done this for a long time, for decades now, theoretically, where we have these sorts of models of how we know that brain development is occurring. We know that patients vary from that in particular ways, and we expect that this is occurring earlier and it's occurring as an alteration of development. But we haven't been exactly identifying. So what I'm proposing is that while it's difficult to look at these normative developmental pathways and identify risk factors, because risk factors can occur and accumulate at any point in time and alter development, instead we know that estrogen is going to be presenting itself within a certain time period for individuals, and it's going to be pushing that sort of developmental alteration back. So if we can figure out where estrogen is engaging and how it's engaging, you might be able to identify novel mechanisms and targets for intervention. So I showed you a bunch of models that are exciting, but I haven't looked at any of those models yet. This is still early in my program research course. I looked at the hippocampus, and the hippocampus vulnerability model is also one that is very important to psychosis research. Theoretically, the hippocampus serves as an interaction point between early susceptibility and environmental stress. Alterations in white matter integrity in the hippocampus predict symptom progression, as does alterations in the shape of geometry. And we see subregions showing a dose-dependent reduction across the psychosis spectrum. with the smallest volumes being in those individuals who have a formal diagnosis, and intermediary volumes in individuals who are in that clinical high risk stage. Estrogen also has several overlapping benefits to the hippocampus. Animal models, we know that there's adult neurogenesis associated with the availability of estrogen. We know that from cell cultures that it increases dendritic spine density and creates synaptogenesis. And in healthy aging models, we see neuroprotective effects on the hippocampus for those who are receiving hormone replacement therapy compared to those who are not. We also see that across the menstrual cycle, it modulates functional plasticity. So if any of you are familiar or think a lot about risk models, what you'll often expect to hear from people studying risk is we had someone who is sort of on this edge of converting, on the spectrum of symptom risk, and their outcome measures are often, hey, did this person convert to a formal diagnosis? That doesn't really work with resiliency work per se, because by its very nature, we're trying to study people who are not converting over to a formal diagnosis. So instead, you'll hear me talk about people who are hanging out sort of in this sort of risk area and not moving into a formal diagnosis. So you'll hear me compare groups that are clinical high risk for psychosis to those who are healthy matched peers. And you'll also hear me talk about psychotic-like experiences. And these are those very low-level, unusual beliefs that I mentioned. So for the first study, we had 60 female participants. 26 of them were clinical high risk for psychosis, meaning that they had those attenuated symptoms that don't quite meet intensity or severity or conviction criteria, and 34 healthy peers. They were 12 to 21 years old, so they were on sort of that high end for transition from adolescence to early adulthood. They had a wide range of age at menarche from 11 to 16. And they varied in time since menarche, with some people having 0 years since menarche and others have. So we were able to look at relationships between age at menarche, time since menarche, to get a sense of whether this was a timing issue or a duration of exposure benefit. And what we found is that early timing of menarche in a whole brain analysis related to more normalized hippocampal connectivity to the occipital lobe. In other words, CHR, so clinical high risk for psychosis females, had hippocampal connectivity that looked more like their unaffected peers if they had earlier timing of menarche. And we didn't see any evidence for relationship to time since menarche, suggesting that it's the timing of this occurrence and not the duration since occurrence that the benefit is. But this is all retrospective. So we also wanted to look within this.
Just quick clarification.
Yeah.
Why visual?
Because it's what came up in the whole brain analysis. We started with the hippocampus and let it tell us. Yeah, that's a good question though. So we wanted to look at people as they go through this transition from childhood into adolescence, to look contemporaneously at this sort of neurodevelopment. So we use the ABCD sample. It's 21 sites across the country. It's a broad geographic, demographic range. And we had 4,422 female adolescents. At the time that the data was pulled, they varied each from 8.9, so almost 9 to 13 years old. And they had varying statuses. Now, remember that this is kind of early for a lot of people to be transitioned menarche at that lower end, 8 years old. So we have 2 time points. For some people, they were pre-menarche at both of those time points, about 2/3 of the sample. 1/3 of the sample had a 1 time point that was pre-menarche, and then at the second time point 2 years later, they were post-menarche. And for about 6% of the sample, they had their first menses prior to 9 years old, which is we looked at their ratings of psychotic-like experiences as their symptom measure outcome. And for the R21 that's cited right there, we took a quick look at just this subgroup, 1/3 of the sample that had pre and post. And we saw that PLEs were reduced from pre to post. And we saw hippocampal volume increased from pre to post. So we thought that this was a good initial indication that this might be a useful signal. So we continued to look using this old model that we found. So hippocampal disconnectivity, we expected in pre-menarche to relate to increased symptoms of PLEs, meaning that maybe this is a biomarker that would be relevant. Then we see that during menarche or after menarche, estrogen would reduce this hippocampal disconnectivity that we see, and that hopefully this would relate to reduced PLE severity. So what we found was bilateral hippocampal volume to visual networks based on the whole brain analyses did relate to reduced hippocampal connectivity. And we see this is true also in our pre-menarche group at both times. In the post-menarche group, we see no relationship to between hippocampal disconnectivity and symptom severity. And then our pre to post, we see a change in direction. When we look at the mediation of this effect of the interaction, we see that hippocampal visual network connectivity no longer significantly contributes to the model with this when this interaction term is introduced. But we do see that menarche still contributes. So kind of not exactly what we expected, but some of this is in line with what we expected. We thought that with menarche would reduce the importance of hippocampal disconnectivity to PLE symptoms because it would be reducing that one pathway. But this is one pathway that would be mitigated. But we saw something a little bit unusual and unexpected. In the earlier study, we said that earlier was better. But here we see that those people who are individuals who are post-menarche at baseline, so those were people who had menarche before age 9, had the highest PLE ratings, followed by the individuals who are a little bit early in the pre-post, and then the lowest symptom ratings, those who are free. So this isn't what we expected, and this isn't what previous literature had found. They had found earlier was better, except that earlier in those studies was more normative. So when we get out of that normative timing in this very early sample with a wide array of first menstruation age, we see that post-menarche around age 8 or 9 is higher risk again. And then as we get more normative, then we see that PLE symptoms are low. So this is an update of the previous finding. Earlier may not be better, but there may be sort of a more normative window in which estrogen may be conferring this effect. And hot from the presses, this updated version, we do see that if we take the age at menarche and we do a Z transform with taking the absolute value, so not caring about direction, but seeing how far away from the most normative value it is, we do see that people with the highest level of symptom severity also have the broadest range of agent. And I should note that this is in a limited range because we only have a little bit past age 13 here. And so we don't have the full array. Not everyone has transition. So what's so interesting about timing, right? Because we say that it's about 12 years old. So 12 years old is actually around an interesting neurodevelopmental transition period where we have peak velocity start to shift for gray matter development, white matter development, subcortical volumes. We also see an intense maturation of functional networks. And if you'll look at the X value here, we have, this is 11.6, 12, 12.4. It's a very condensed time period where we see these networks start to coalesce. And this was measured as a ratio of within network coherence versus between network coherence, as a sort of sign that these networks are now forming and becoming more distinct. Whereas in childhood, there is way more between network activity. So these are some exciting future directions of how and where this might be occurring. It may be impacting these neuromaturation effects. And I'm getting close to closing, so I just want to start to do some conceptual tying up by saying these effects may not be the answer. This may not be the solution. I do think that if we had a treatment that consistently delayed psychosis for 5 to 14 years and supported role and social functioning for individuals over the course of psychosis and improved clinical course, we would be getting MacArthur Genius grants and high-fiving. And we do. It's normative female development or something like it or something associated with it. So I want to just emphasize that there is an enormous amount of potential, I think, for us to identify routes, help, not just men, but men and women have a better clinical course. So understanding how these normative changes may impact mechanisms of psychosis, they give us critical insights into where, how, and when we can reduce symptoms or delay onset as a part of a large array of treatment approaches. So how would this potentially look? Well, I think that there are several ways that we can do this. Yes, we can give estrogen directly to females with psychosis, especially surrounding vulnerable windows. Recent research has shown that this is actually helpful in working to reduce the occurrence in individuals who are at risk in postpartum and premenopause treatments. But we could make estrogenic type effects for all patients using things like exercise to improve hippocampal connectivity. And we've seen that high impact interval training exercise does seem to act on hippocampal connectivity to visual cortex in a similar way that estrogen was. Why? I'm not sure that that's a selective effect. But we also see benefits of neuromodulation to the cerebellum to help support normalizing connectivity. And this is also seems to be occurring in similar fashion to estrogen. I'm going to talk about this a little bit more broadly. I think in the original talk title, I promised psychopathology and not just psychosis. And, as many researchers are, I was a little bit overly ambitious about how much I would get done between now and then. But I will say that I do think that there is a great amount of potential to apply this to a broader number of disorders. 75% of first onsets of psychopathology happen in adolescence. There's a lot of normative female neurodevelopment that may have distinct effects and result in sex differences for many disorders. For example, bipolar disorder is more likely to present first with mania, and women are more likely to receive depression diagnosis and have a delayed onset of their first mania. And depression is more prevalent in women, but also may be more prevalent in disorders. So it may not be, estrogen is not necessarily beneficial to all forms and all settings. We saw that even within psychosis, women had more severe depression. So it's not a complete sunshine and rainbows sort of solution, but it's complicated and may have a potential to understand it. All right. So thank you so much for giving me this platform to talk about these ideas and this potential. All this work was supported by NIH and from the families and scientists at the ADAPT lab at Northwestern and ABCD all across the country.
Thanks for that. I'm just curious about the general symptoms of psychosis. Like, what is the example of symptoms?
So PLEs are psychotic-like experiences, which at age 9 to 13, you aren't having in the normal course or consideration of like psychosis development, you're not having sort of these attenuated symptoms yet. You get questions about things like people will come in and they'll say that I have special knowledge or superpowers that my friends don't have, or I have a feeling that I might be able to control things with my mind. But again, these are things that you're like, are you sure? Or like, I don't know, I like to think this sometimes. And then it does predict progression of symptoms in the future, but it's not the same sort of measures that you would use in someone who is in late adolescence, because they are more likely then to have these sort of more formalized symptoms with that sound more psychotic-like than these very early.
This research, there's also people related to testing.
Yeah. So default mode network, there's an interesting finding using normative correlational distance. So looking at default mode structure across a large number of individuals and seeing how different that is, your connectivity is from normal connectivity. And you find that people with psychosis are actually can be classified based on that because they tend to be the farthest, with bipolar disorder being nearby folks that are slightly closer to normative default mode network structure, and then depression being like a third place distance. So it's definitely an important area, and since disconnectivity writ large has been sort of a broad neurobiological marker of psychosis, I think that it's definitely.
Yeah. So I'm interested in the connectivity between hippocampus and individual regions.
Yeah.
So have you, or are you going to look at if the connectivity related to their symptoms, for example, the visual delusion, because they have those symptoms, so they show those connectivity between these two.
So we're finding this signal even in folks that, I think that your question is like visual. You think it's delusions, or were you thinking visual?
Yeah, I think maybe like they have those visual delusions, so that's why they have the connectivity.
So the findings both in the ABCD sample as well as the ADAPT lab sample didn't restrict the types of positive solutions to or positive symptoms to be just these visual hallucinations. It included a wide array, and the signal seems to be there even in individuals who are not. Because, this was a reviewer question, take out everyone with visual hallucinations. Do you still see this effect? And we do. And so this has been kind of an interesting thing because it's not maybe primary visual, but hippocampal to visual cortex has also been associated among a larger network with sort of this reality maintenance of like keeping in mind your surroundings and working memory of what is happening right now. And so having less connectivity in that way may actually, so that there's some hypotheses that this might be related to like your brain then fills in what it's not getting in these sorts of primary input. And so that could be a way that you might get a lot of different manifestations of filling that knowledge gap.
So kind of continue a little bit on that.
Yeah.
So you said really nice hypothesis that like center on the hippocampus, right? So there's a good reason to look for the hippocampus. Then it end up being with visual cortex. It's interesting to think how. If you go for prefrontal cortex to even the visual or other region, like if you kind of open it up and perhaps leave the hippocampus as your seed kind of on the sideline, what would you expect, or would you find interesting reasons? Because in your hypothesis, like a lot of other potential hotspots for where to pick on, right?
Yeah. So there are a lot of models, and I do think a lot of these models overlap with like people's favorite pet regions. Cerebellar people like cognitive dysmetria because it makes cerebellum like one of the stars of the show. I'm biased towards hippocampus. I really like it. I think it does a lot of interesting cognitive things. But a lot of them do. A lot of those things are also regulated by the cerebellum. So, but I do. So you were asking if I said not hippocampus, but everywhere else. Part of what I think that we're at risk at is that there is some possibility that schizophrenia, because it's so clinically heterogeneous and because it's so pathway heterogeneous, that you would find sort of an unhelpful answer of yes in everything. And I don't think that that's a wrong answer. I think that you need to have a sample that's sufficient to understand that it may be a lot of regions that play or that are resulting in sort of similar behavioral manifestations, and that there are a lot of pathways that could be relevant for the individual. So I think that the answer is that it's an important thing to expand beyond these sorts of individualist or individual models. But I think that we'll need to do so in large samples with precision level connectivity so that we can have a good, reliable sample to look at something. I think that would be the answer, is that yes, there's going to be a lot more regions involved, but that I think the degree to which a region is relevant for a single individual is going to require us to increase like the precision of our energy.
Just was going to say, it's a shame. Oh, sorry.
You're okay, go ahead.
It's a shame Elena isn't here. You can ask this, make this point better, but to your point, the notion that schizophrenia is a single phenotype,
yeah,
I think is probably incorrect. Seems to be. Therefore, the idea that you find a biomarker, a model that is going to explain psychosis in general is probably a bit of a fool's errand. Sorry, I'm not accusing you of being a fool.
No.
But, so
I think that what I just said was very consistent with that.
Yeah. So in other words, the idea is there's a common phenotype that's driven by many different mechanisms. So Elena and Carol Tominga folks at UT Southwestern have come up with this kind of biotype modeling of psychosis that breaks down subgroups into three main subgroups: they do not correspond at all to classical DSM type breaking down of psychosis. So people from different DSM categories fall in the same biotype. And in some of the work that I've been peripherally involved in, where we use machine learning classifiers, you can do a better job classifying the basis of grey matter concentration across the cortex in terms of these biotypes than you can in terms of more conventional clinical categorizations. So my question is, when you're the research that you do, and do you think that the role of estrogen or hormonal influences more generally might be relevant to certain subgroups of people with psychosis than others?
Yeah.
In other words, if you had a big enough sample to find that these effects that you're seeing, for example, peripubertal, are only impacting the subgroup of people who go on to develop psychosis. Subgroup of females.
So I am not conceptualizing it that way per se. I do largely agree with the suggestion, but the way that I would slightly reframe it is that I think that these are normative. So I do think that the benefit or the impact on neurodevelopment is general. I only think that the benefit in terms of symptoms is going to be enough to overcome risk burden in some instances. And I think it probably depends on where and how that risk is accumulating. So for what regions, what mechanisms, and the types. I do think that there's likely some difference in type of burden. So whether it's stress from obstetric complications, adolescent developmental stresses, or if it's highly genetic. So women who have sort of these highly genetic forms of psychosis where they have high family burden as well as substantial genetic burden seem to show less variability related to estrogen. So I think that there's already some evidence that the type of risk factors matter, that while the benefit is there for, it seems to be there for everyone generally, it may not be enough for certain individuals. Or,
incredible talk. Thank you so much.
Thank you.
Psychosis is a really big interest of mine, and so it's very refreshing to hear some theories on it. But I was wondering, based on your current research, what changes would you expect to see in the onset of psychosis in people undergoing hormone replacement therapy for gender dysphoria?
that is an incredibly interesting question. And something that I proposed to look at in ABCD, unfortunately, I think that they have like five individuals that are undergoing. So it's not a lot that we have. But there is some sort of, I think it would be difficult to disentangle sincere gender dysphoria from psychosis delusion in the way that we've set up our measures. And here's why. I think that theoretically and clinically, they're very, very different things. But I think that the way that we've designed our rigorous research measures, it's very, it's very difficult, because what you see in psychosis is there's a large amount of what they call disorganized symptoms, where we talk about things like, does your body somehow not feel like your own? Does it cause you distress? Typically with people who have psychosis, we could, you hear something from them like, my arm's being controlled by the CIA. Or, this is not my arm, it's someone else's arm. It's very distressing. It's very upsetting. My arm looks and feels different. And those types of delusions get wrapped up in sort of these gendered dissolutions where, I also feel like my body is not my own and it's different and it does not, belong to me or reflect me. And so I think that it would be very, very hard to disentangle that from the sincere therapeutic effects of, actually having gender affirming therapy and having your body now match these sorts of changes. Because your distress is going to go down. You're going to say your body feels more like your own. And I don't know that those individuals necessarily will rate other places and heal. That makes sense?
Yes.
Yeah. Okay. It's hard to disentangle, but I do think it's a fascinating question. I also think it's a fascinating question for people who are maybe going through puberty extremely quickly, where, you wake up one day and grown 3 inches, or, your body feels legitimately very different than it used to. And that is currently rating highly on PLEs. Whether that actually is impacting your sense of self in a way that's psychologically relevant, where suddenly you feel like you don't have a strong sense of self, or whether it's actually just reflecting a real stressful change, I think is a relevant question. We run into this in absolutely every other area of psychosis as well. I'll just note that that's not just bodily changes, but we see this with people having paranoia. Paranoia is higher in individual areas where crime rates are extremely high. But then is that legitimate paranoia that you should be fearing for your life because you're in an area where you may be involved in a violent act of crime? And how do you parse that from disproportional anxiety about your safety, or real, what we would want to call paranoia, where it's you have nothing, but what if you do have something to do? And I don't think that we have, or what is the appropriate level of fear that you should, in the stress that you should feel about living in a neighborhood that way. I think that that's something that we really have to think about and grapple with.
Just follow that. So you pre-staged your comments with some ideas about environmental exposures.
Yeah.
And there's the idea that early childhood adversity leads to accelerated onsets of puberty, greater sense of PLEs, and also what would look like more connectivity. The idea being that there's earlier maturation than you would expect, which might be a bad thing. So have you incorporated those measures, especially in the ABCD sample, because it's almost going in the opposite direction of your estrogen.
Right. Yeah. So that is something that's currently in progress. I have looked at it without including puberty lens to it, and I have seen that we do see this relationship between ACEs, hippocampal development and connectivity, and PLEs, psychosis risk symptoms. And I am really interested in this question because this is something that's biologically preserved across, from grasshoppers to humans. This high stress environment changes your pubertal development. And it's an interesting question because if we have normative timing is better, then is this a short-term gain for a long-term loss? Because it is feeding benefits. It may, there's some suggestion that it may be counteracting this sort of sustained high cortisol exposure that might be deleterious, but estrogen and early pubertal development might be counteracting that. But it is also potentially maturing networks that are not ready to be matured or reorganized, and maybe causing some long-term effects. And I think that's a very interesting question. This is why I'm also excited to look at it in ABCD, where we might be able to parse out some of these subgroups, because we'll have a large enough sample to look at high ACEs exposure versus low ACEs exposure, and early versus late puberty. So
because you could. I appreciate the there's a lot of content as you highlighted on anything else, but it would be interesting to see amongst those kids if you equate for ACEs and then look at boys versus girls. This earlier menarche actually lead to protection for that specific group by ACEs relative to the comparable requirements.
Yeah, and So there is a benefit of early pubertal development to males as well. And part of that is testosterone has like benefits to dopamine sensitivity. And so you might be seeing benefits that are differential, but also potentially like overlapping in some ways. If you have more sensitive striatal dopamine response because you had early male pubertal development, women aren't necessarily receiving that same benefit, which may have implications for depression, but also clearly have implications for mania. So I think that this is, I think that it's a really complicated question, but I think it's important to dive in.
Great. You want to ask a question? No, I was just going to ask.
For online. So your findings are quite reminiscent of findings we had presented to us at our Ziotis lecture last year that showed a very similar effect of age of onset and rapidity of onset of menopause and Alzheimer's risk improvement. It's a fascinating point of comparison and sort of begs the question of maybe there's some sort of common mechanism between these two very distinct processes. But that in addition to your comments about this being a question of differences from normative change, I think suggests an interpretation of your findings that rather than estrogen being a protective factor, it's the change in estrogen being a threat to homeostasis or a homeostatic insult that could be causing these disruptions in a number of complex processes, in this case, expression of psychosis, expression of Alzheimer's disease. How do you disentangle the estrogen as a protective factor versus the homeostatic shift of these periods as a detrimental factor?
So I think that there are a couple layers to this. And the way that I don't think that those need to necessarily be conceptualized, but I will say that I think that there's two layers. There's an acute layer, and then I think that there's like more chronic neurodevelopmental layer. And so I think that these large changes are likely impacting these healthy aging and healthy adolescent development factors in a broader way. So you have these huge changes. We see in a recent paper by Emily Jacobs, where she scanned someone over the course of their entire pregnancy, that there's a lot of neural reorganization that happens in this matrescence period as well. And so I think that with those huge gaps, we are seeing large-scale neurodevelopmental changes. And I do think that those probably have some staying. We also see menstrual cycle related changes. And I think that those are likely having acute functional effects instead of like large scale network changes. These are just hypotheses. And so in some sense, you're talking about, okay, we're removing estrogen, or these changes in estrogen are, it's not just estrogen being a protective effect. It's, maybe the removal or a large scale change. I think both probably, because we see these acute impact factors as well as these large scale changes.
So it sounds like your perspective is that we're looking at the aggregate of separate effects caused by this.
Some of them are probably neuromaturational. Some of them are acute benefits.
In the interest of time, are there any questions online?
There are no questions online.
Thank you.